Hello everyone. In this video series, I have already discussed about the biography of Gregor Johann Mendel, then monohybrid cross which Mendel performed on garden pea plant. In this type of a cross, he studied inheritance pattern of single character from parent to progeny, and on the basis of results of monohybrid cross, Mendel proposed two different laws which is called as first law of inheritance and second law of inheritance, or law of dominance and law of segregation. Then I also discuss about dihybrid cross which Mendel performed, and in this type of a cross he studied inheritance pattern of two character at a time, and on the basis of dihybrid cross findings Mendel proposed third law of inheritance which is also called as law of independent assortment. Now in this video I am going to talk about last experiment that Mendel performed, and this is nothing but the test cross. Okay, so in this video we are going to discuss test cross in detail. Okay, so it is the simple type of a cross developed by Mendel and the main aim or the main purpose of test cross is to find genotype of unknown organism. Okay, suppose we have an organism and we want to determine its genotype. In that case we can use test cross which Mendel performed. Actually Mendel did test cross on garden pea plant and he performed test cross on F1 hybrid plants. Okay, so we know that in monohybrid or in dihybrid cross, after parent cross, the first generation which is called as F1 hybrid, and Mendel used this test cross to find out the genotype of this F1 hybrid plant or first generation plant. For example, if you are walking on the road and you find two garden pea plants, one is dwarf and another is tall. So in that case, you can easily determine the genotype, or you can easily tell genotype of the dwarf plant as a small t. Okay, this is the phenotype of the plant, and we know that dwarf is a recessive character which is present in the garden pea plant, which is controlled by small t genes. So its genotype always will be small t, but it is not possible to determine genotype of a tall plant, because tall plant may have genotype capital T, small t, or capital T, because both this type of a genotype produce phenotype tall. Okay, and this will going to create a problem, and to solve this puzzle Mendel developed test cross. Okay, so it is the simple cross developed by the Mendel, and the main aim of the cross is to find out the genotype of unknown plant or unknown organism. Actually he performed test cross on F1 hybrid or first generation plant which he obtained in monohybrid and dihybrid crosses, because we can easily talk about the genotype of a dwarf plant because it is small t, but it is not possible to tell genotype of a tall plant with a naked eyes, because it may be capital T or capital T, small t. Okay, and here test cross come in the picture. In test cross, Mendel cross F1 hybrid plant with homozygous recessive parent. Okay, so this test cross Mendel performed in between first generation plant or F1 hybrid plant and recessive parent. To understand this, we have to draw monohybrid cross of the Mendel. For example, in monohybrid cross, Mendel take pure tall plant with a genotype capital T as a one parent and pure dwarf plant with the genotype small t as another parent. Okay, and then Mendel perform artificial cross in between these two tall and dwarf plant. It is the parent generation. We know that the pure tall plant produce gamete capital T, and this dwarf plant will going to produce gamete small t. So these are the gamete. They produce single type of a gamete, and after artificial cross, these two gamete combine in F1 generation or in F1 hybrid. So the genotype of the F1 generation become capital T small t, and due to the dominance of the capital T, the plant become tall. That means this F1 hybrid show phenotype tall and its genotype is capital T small t. And if we want to determine the genotype of this tall plant, we have to cross this F1 hybrid plant with one of the parent. Okay, this is the parent of F1 hybrid plant, and Mendel select homozygous recessive parent. Okay, this is homozygous dominant parent, and this is homozygous recessive parent. So Mendel cross F1 hybrid with homozygous recessive parent, and this type of a cross is called as test cross. The cross between F1 hybrid and homozygous recessive parent. Okay, now as we cross F1 generation plant with their parent, it is also called as back cross. Okay, as we are going back in the generation, we are crossing F1 progeny with their parents, one of the parents, so it is also called as back cross. Okay, now what is the result of this cross which Mendel performed? This capital T small t type of a F1 hybrid produce two type of a gamete, capital T and small t, while this dwarf plant in the cross produce single type of a gamete which is capital T. So I am writing this small t genotype of the dwarf parent here. It will going to produce gamete small t and small t. So if we make Punnett square, we can easily calculate the genotype of progeny in the test cross. Okay, so here this capital T small t and capital T small t plant will going to give phenotype tall. Okay, they will going to become tall. Now small t and small t plant will going to show dwarf phenotype. Okay, so out of the 4 plant, 2 will be hybrid tall and 2 will be the dwarf, which is a recessive phenotype. So the ratio of test cross become 2:2. 2 are tall and 2 are dwarf. Okay, so we can write 1:1. Instead of this 2:2, we can write 1:1, and this is the test cross ratio. Many time in competitive exam, they ask about the test cross ratio, which always will be 1:1. If the F1 hybrid is showing capital T and small t genotype, then the ratio will going to become 1:1. Okay, so we have to remember different ratios now. Monohybrid cross show phenotypic ratio 3:1, while dihybrid cross show phenotypic ratio 9:3:3:1. Now the test cross, which is the cross between F1 hybrid and dwarf parent, going to show phenotypic ratio 2:2, or we can write 1:1, because we are going to get 50% tall and 50% dwarf plant. Now, if for example we cross this F1 hybrid with dominant parent, okay, now this type of a cross is called as back cross. Okay, we are going back in the generations, and in this type of a cross also we are going to get a ratio, but in this cross we are unable to make any conclusion about the genotype of F1 hybrid. For example, now this capital T small t will going to produce same gamete, capital T and small t, and this dominant parent will going to produce single type of a gamete, which is capital T. Now if we make a Punnett square here, it will going to give capital T and capital T, both are tall. In this case it is capital T small t and capital T small t, they are also tall. So when we cross F1 hybrid with a dominant parent, in that case ratio will going to become 4:0. All are tall, 4 tall, 0 dwarf. Okay, and out of this 4 tall, 50% are pure tall and 50% are hybrid tall. Okay, so this is the back cross. But by using this cross, if we cross F1 hybrid with the dominant parent, we cannot make any conclusion about the genotype of F1 hybrid, because all plant will going to appear tall. So we have to cross the F1 hybrid with recessive parent. Okay, it is called as test cross. So on the basis of now this cross, we can make one very important sentence or conclusion here, is that all test cross are back cross, but all back crosses are not test cross. Why we are saying like this, that all test cross are back cross? Because if we want to test the genotype of F1 hybrid, we have to go back. We have to perform back cross. Okay, so all type of a test cross are the back cross, but all back cross are not test cross, because if we back cross F1 hybrid with the dominating parent, in that case we will not going to get any solid conclusion. So in that case this back cross will not going to become test cross. So all test cross are back cross, but all back crosses are not test cross. Okay, Mendel also used same type of a test cross to determine genotype of dihybrid F1 generation. Okay, because in dihybrid cross the F1 hybrid show genotype, for example yellow and round parent when crossed with green and wrinkle parent, they will going to give F1 generation as capital Y small y and capital R small r. Okay, this is the F1 hybrid in dihybrid cross. If we use similar method and Punnett square, you can easily determine the different type of gametes produced by this F1 hybrid, and when we cross this plant with their recessive parent, that is small y, small r, we can easily make the ratio of dihybrid cross or dihybrid ratio, and it is 1:2, 1:2, 1:1. Okay, so here when we use test cross in monohybrid cases, the ratio become 1:n, And when we use this test cross in dihybrid cross experiment, the ratio become 1:2, 1:2, 1:1. Okay, you can easily determine it with the help of Punnett square. Thank you.